Hi, this is Miss Litton. This is my wonderful honors bio class. Say hi. Hi. Period 4. So what we're going to do right now is I'm going to introduce the overall lab to you. It's going to serve two purposes. One, you'll understand the lab better. But by doing this today, your lab prep for Friday is not a difficult lab at all. I'll show you that. The prep is very, very simple. But once we start in on the real deal lab, that lab map in your notebook is due on Tuesday of next week. That one is more complex, but if I show you what we just learned moments ago in chapter 24 and how it relates to this lab, it'll make doing that lab write-up, I want to say 100 times easier. Okay? So you want to understand this because you could get this done if you have a busy weekend. You could prep for this lab, and when we finish talking about it, you could prep for the bigger lab next week starting today. So you can get it done and you're ready to go for class on Tuesday. All right, so there's some things in here you should recognize already on this picture. Where are some things you recognize in this picture? Sharpies. Okay, good, Sharpies. Used by all good genetic engineers. What else? Yeah?
I recognize those. Isn't that like the bin where you put the micropipette tips?
The tips right here. Never dip without a fresh tip, okay, with your micropipettes. Good tip, okay? What else do you recognize in here? Do you remember these blocks right here to set your microfiche tubes? These right here are my stock boxes. You will have your stuff that's there, but every day, obviously numbered because you know, okay? You will have a stock box that has the solutions you need for that day's lab. Will be up here for you to come and get and then return to me at the end. This is what's gonna hold your, remember how you eject the tips on the side? It's gonna hold that. So you have other things as well, and we'll go over the details of what's in your bin over there on Friday when we go through the lab. I'm just kind of going through the processes with you now. So the first thing I want to do is see how much you remember from what we just discussed. So here is a quick little quiz, not a real one. So we can see how much you remember. I'm not going to give you very much time because I expect you to whip through it. Are you good? Finish up, finish up, finish up. Let's go, sugar phosphate. Let's go, I don't know what you are. Yay! Did it give you a score?
No. Well, I got it.
Oh! How can we guide these two? Microbes are just anything, eyes to me, right here. Microbes are just anything that are what? Tiny, small. Okay, so this is saying that are prokaryotic without organelles, that would be bacteria. But most of you got that. Meh. Small rings of DNA, they're called plasmids. Somebody said vacuoles. Bacteria don't have vacuoles because that's an organelle. Really?
No.
Okay, what do you use to tape? Ligase. Okay, somebody pick DNA polymerase. What is DNA? Okay. Somebody pick PCR. What's PCR? Polymerase chain reaction. Use that to do what to your DNA? Photocopy it. Okay. Oh my gosh. Okay, what do you use to separate out the pieces? We do gel electrophoresis. Two of you said PCR. What's PCR for? Photocopying the DNA, making more. And yay, you all got that. Good job. All right, so let me talk to you about this. Okay, do me a favor and just for a minute, kind of lower those Chromebooks so I can get your attention up here. I will help you do everything that you need to do. You'll understand it, okay? Just need to, hi, sweetheart, how are you? Good. When convenient, okay. I want you to go at the end. All right, now, no stress, no stress. Get your story straight, you're right next to each other. Okay, now, I'm just saying. All right, so here's the deal. Here's the bacterium, here's an antibiotic. Obviously not drawn to scale or reality in any way. But antibiotics are set to do what to bacteria? They're trying to what it? Kill it, okay? Bacteria, some of them have genes that code for proteins that help them if an antibiotic comes along, they will live because they have the DNA that will protect them against that antibiotic. Now, as I mentioned earlier, you will have two plasmids that you're going to work with. Eyes up, okay? Focus. You will have two plasmids. Here's one of them. Don't worry about the notes. I'll help you fill it in just a minute. This is the first one called pecan, and it's actually pecan-R. The R stands for recombinant. So this is a genetically engineered plasmid, and it is sold for the very purposes of what we're using it for. Pecan-R, it says 5512BP. Any guesses what that might mean? Yes. Good call. Wonderful. Okay? It means it has 5,512 base pairs. Remember what base pairs are? That would be A with T and C with G, right? So plasmids are rings of DNA, and this ring has 5,512 base pairs in it. Now on it, there are several different genes, but one that I want to draw your attention to right here is CAN-R. That stands for kanamycin resistance, which means if this particular plasmid, PRR, is inside of a bacterium, if there was kanamycin here, that antibiotic, he would live because he has the gene that codes for the proteins that will protect him against kanamycin. Will he be protected against penicillin? No. Amoxicillin? No. Ampicillin? No. But he is protected against kanamycin. He has some other genes on there too. I'm going to revisit those in just a minute. The second plasmid you're going to be working with, okay, is Piara. Now, is it bigger or smaller than pecan? Small. How do you know? Yeah, it has fewer base pairs. You're up here, Courtney. You're focused here. Okay? Now, this one does not have kanamycin resistance. This one instead has ampicillin resistance. So if this plasmid is inside a bacterium, they will survive even if ampicillin is around. Now I want you to look at something else. Do you see where it says Hindi and BAM? There. Do you see the BAM Hindi? Those are recognition sequences for the restriction enzyme BAM and the restriction enzyme Hindi. So that it can do what? Cut and create what? Sticky ends. Okay. This right up here where it says ARA-C, ARA stands for arabinose. When arabinose is present, it will help RNA polymerase bind to the promoter if arabinose is present. Okay, it will help trigger that process. Now here are the two plasmids side by side. Did I draw them correctly, size-wise? No. No. Why? PR should be what? Smaller. Do they both have the same recognition sequences? Remember what recognition sequences are? Where restriction enzymes cut. What were the two restriction enzymes I told you we would use? BAM and Hindi. Do they both have BAM Hindi sites? Do you see them? Tell your bio buddy where the BAM Hindi sites are. Okay, now, here's BAM, here's Hindi, here's BAM, here's Hindi. Now, here's kanamycin resistance. This says RFP. This stands for red fluorescent protein. This is a eukaryotic gene that was once housed in jellyfish. Next to it right here is PBAD. You know what PBAD is? RNA polymerase binds to a named PBAD. However, he has nothing to help him. So because there's nobody to help him, this red fluorescent protein never gets transcribed and translated, so the bacteria never glows red. I wonder where we could put him. Try to anticipate what comes next and tell your bio buddy, what could we do? Look at the restriction enzymes. What could we do to turn on this red fluorescent protein? Talk about it with your bio buddy. Now let me see if this is what you said. Did you say to cut here and here and cut here and here?
Yeah.
And you would cut once with BAM and second cut with Hindi, and then you have 2 fragments here and 2 fragments here? And then did you say to take that fragment and sneak it in over here? Because then we take this out of here and replace that other plain old fragment, replace it with this. Now, if we grow this bacteria, if we put this in a bacterium, right now it's just a thumb drive, now it's just DNA. But if we took this plasmid and put it into a bacterium and fed it arabinose sugar, the arabinose would help RNA polymerase bind to the promoter named, which would turn on then the RFP gene, and that bacteria, not only would it be resistant to ampicillin, it would also be what color? Red. That is your lab. Okay? So what's going to happen is Friday's practice. Tuesday next week, that's the cut day. Thursday next week, that's the ligase day. The following Monday, that's when you're going to do gel electrophoresis to see if you cut and taped. Then the day after that, you're going to be God, your play God day, you're going to do transformation. You're going to force your bacteria to absorb your new plasmid, plate it out, and see if it glows red or not. Okay, that's the sequence of events. Now, take a look at your notes on this. Okay, the plasmids. PCAN-R, I've given you the base pairs. CAN-R is a gene that makes bacteria resistant to the antibiotic kanamycin. CAN-R is the gene that makes bacteria resistant to the antibiotic kanamycin. PBAD is the, say it, promoter. PBAD is the promoter sequence for transcription of a specific gene. RFP, those are genes that code for, what's the word there? Red fluorescent proteins. So that's your PCAN over here on the left. PRA over here on the right, it has ampicillin resistance, a gene, AMP-R, a gene that makes bacteria resistant to the antibiotic ampicillin. It has ARA-C, which activates neighboring promoter. And who's our promoter? PBAD. That's what we want to make. When bacteria is grown in the presence of arabinose. Arabinose is a sugar. ARABINOSE. Arabinose. Basically acts like a switch to turn on a gene. Our restriction enzymes. We got BAM and we got Hindi. Each what? Cut. Each cut at a different recognition sequence. Both plasmids have recognition sequences for the enzymes. Why 2 sites? I've given you the introduction to that, but look up here. This is big time thinking. Why couldn't I just have BAM, BAM, and BAM, BAM? Why did I need 2 different restriction enzymes? Yes?
There's one restriction enzyme performs one task.
It is, but they could have genetically engineered this so it was BAM, BAM-ish.
Because if you had the same one, you wouldn't know which way to actually enter it.
Yes. And I'm going to tell you, this lab has evolved over the years. When this lab first started, the only thing that was done is you put in ampicillin resistance. That was it. That was like huge. We're like, well, this bacteria grows on the antibiotic. This one doesn't. Wow. And they do that to select And that way you know the only ones living there have that gene and whatever gene you put next to it. So it eliminates all the ones that haven't been transformed. That was the original purpose of the lab, just to show that much. Then came this step, and at first we only had, we had the same enzyme. It was actually called CYP1. I remember its name. And it was, we had a CYP1 cut here and a CYP1 cut here. But if you did that, then it could go in this way or go in the other way. And you needed to have ARA-C right next to PBAD. So that's why two different ones. And also it prevents it from closing in on itself and recircularizing with itself once you cut it. Because if they had a CYP end and a CYP end, it would just, sticky ends would match. So as soon as we cut it, it would go right back together. So on your notes, why choose sites? It ensures that ligated DNA is oriented in the correct position. In the correct position. And it prevents the plasmid from reforming without inserted gene. Without inserted gene. So steps of the lab, lab 1, on Friday we're going to learn how to use the micropipette and gel electrophoresis. Lab 2, digesting pecan and piara using the enzyme to cut both plasmids. Lab 3, ligation, building the piara-R plasmid. You are combining fragments of pecan and piara to form a new plasmid. Combining fragments of pecan and piara to form a new plasmid. Okay, then the following class period, we're going to do gel electrophoresis. Did we cut it? Did we cut it in the right spot? Did we form and ligate a new plasmid? You're going to get a gel that looks somewhat like this, but your large mammalian brain is already going to understand this part, and it will help you when you set up your lab. Across the top in this well hole right here, you're going to have loaded in marker. Known pieces of DNA. Remember when I talked about that earlier? And it's 10 fragments. This is 10, 9, 8, 7, 6, and then 5, 4, 3, 2, 1. Right here. Those are known fragment sizes. This right here is your pecan uncut, so you call it K-. It does not have any restriction enzyme in it. But do you see how it has a smear, like a skid mark in there? And that's because whole plasmids sometimes travel like this, but sometimes they twist up and travel faster or partially twisted. So that's why you see the skid mark. Here, okay, this is K+. It has restriction enzyme in it, and you get two distinct banding patterns. Those two distinct banding patterns are this piece and this piece right here. You see that? And that smaller piece would travel farther. Here is piara, A-, you just get the skid marks. Okay? Then this is with the restriction enzyme. So you get this one and that little itty bitty piece out there. So you know you cut it when you compare the uncuts with the cuts. Uncuts with the cuts. You know you cut it in the right spot because you look to see where do the fragments line up to the marker. And then this is your L tube, your taped tube, and it has a bunch of pieces here, nothing down here. Why? They've all been what? Reformed. So they're more the same size as some of our original plasmids. This is how you can see if you cut it in the right place, and ligate it. That is day 4. So the word you want there for gel electrophoresis is verification of digestion. And ligation, lab 3. And then day 5 is your transformation day, your play God day. You're going to force your plasmid into bacteria. And if we put this bacteria on something called luria broth, it'll always grow. Anything can grow on luria broth. But if you put it in something that's got ampicillin in it, it'll kill off any bacteria that don't have the resistance. And then if we put arabinose in here as well, then this whole bacteria will start to glow what? Red. Because it has that gene in there. So this particular plate has ampicillin in it, and it has arabinose on this plate. So this guy right here, he's resistant to ampicillin, but this guy, remember it was one bacteria who made clones of itself, right? This bacteria is resistant to ampicillin, oh sorry, yes, and it also has the RFP gene in it, which makes it glow red due to the arabinose. Oldest bio buddy, this is your slide to explain right here. Go ahead, oldest bio buddy. Not yet. All right, now this is your content, but now let's look at the lab so you're ready to go, because this is a lab. I expect you to get all the points. Go into Google Classroom. Okay, so underneath where it says AMGEN Lab, this right here is a digital copy of the lab book that's at each of the lab benches. So you can use either one you want, I don't care, but when you're home, obviously this would be the one you would use. Now, you will see 3 lab maps here. Let me explain them first here, and then we'll open them up. AMGEN Lab Map 1, this is for this Friday. It is very simple lab map. You're going to open it, you're going to prepare for it, you're going to turn it into me on Friday with your term stapled to it. Okay. This lab map will take place for labs 2 through 4. Lab 2 is the cutting, lab 3 is the taping, and lab 4 is gel electrophoresis. We're going to combine those 3 labs into one lab that you turn in, and all of these go in your lab notebook. And then AMGEN Lab Map B, this is for your God Day and the results. Okay, so we have 3 different turn-ins. Let's look at this one first. Okay, this is what I want you to set up for Friday. Boy, I know it's going to be hard. Hope you can manage it. Hold on to your horses, my friends. Okay. So here you can see this very long driving question. How do I move small amounts of fluids and separate them? Here's your background.
That is so
I know. I'm sorry. I'm hideous. Have this. Your data. Do you remember last year? This is going to be good. Just review practice for you. Remember when you would make little dots onto this? Okay. So this right here is you're going to write drops. You're going to write the names, you know, how much is in this drop in this order. This is not going to be in 2 rows. This is going to be in 1 long row, right? And then you're going to make 5 exact same size circles. Because the point of this is now tell me those circles here represent these circles here. Now I want to see what the drop looks like in relationship to those circles. So a 20 microliter drop might be like this in relation to this circle, but a 5 microliter drop might be like this in relationship to that circle. So on the day of the lab, you're going to show up with this already drawn, but then once you make your drops, you're going to draw on in red, using a red colored pencil from my copy, what your drops look like in relationship to those circles. Have you done that before? Mm-hmm. Yes, so that should be really easy, right? Okay, so that's the first part, and then the second part is you're going to draw a box like this, and it's representing the gel before you load it. These are the little well holes. Remember the little well holes? And if you want to be completely accurate, you can make your gel, you are actually going to have 6 well holes. but you are going to repeat, and you're going to put solution 1, solution 2, solution 3, and then you're going to repeat it again. Okay, so that's really what it's going to look like. If you want to make it like that, you can, or you can just draw one set of it. Okay, and you're going to be loading for practice, you're going to be loading inks in here, and then these inks are going to separate themselves out. Okay, I'm just doing a different one. Oh, poo-poo. These inks are going to separate themselves out. Okay, once you run the gel, and you're going to draw what that looks like when you're done. And that's why I say draw bands that you see, name the colors that you see here. And then you have one conclusion question to answer once you've done the lab. Which die is the smallest? Support your answer with evidence. Okay, that's Friday's lab. Come to class on Friday with this all ready to go to receive those results. That is easy peasy. You're going to turn it in with your terms. What is a little more involved that I want to show you, go to this one. This is what's due on Tuesday. I want to go over this with you in case you want to work on it ahead of time. And it looks actually much scarier than what it is in reality. Okay, so let's make sure we understand it. Get your eyes up here or look on your Google Classroom if you want to. It says we're going to be doing this lab over 3 class periods. The driving question, the background, the hypothesis, and flow chart just for lab 2 need to be in your lab notebook by when? 3/28. That's next Tuesday. The rest you will add as we go along. You'll add in lab 3 by 3:30 and lab 4 by April 3rd. Okay? And then you'll have a different one. The last of those 3, that's going to be due on 4/5. Now, the driving, I've given you the title. Here's the driving question. Look at the background. Okay, I've done them in 3 different colors, though these are all due the first day, just to help you differentiate the 3 different labs. So it says, from lab 2, explain what a plasmid is. Where can you find the answer to that? B4. I want you to get it out. I want you to have the different voice, so I want you to get it from the lab book. If you don't have a lab book at home, if you're not doing it here, where could you find that information? On the digital lab book. Could you work on this if you finish your lab early on Friday? Yes. Okay. So I want you to explain what a plasmid is using B4 of their lab book because I want you to have a different voice. Okay. Can I get eyes up here because some of you are looking at other things? I'm not sure. From lab 2, explain what a restriction enzyme. Could you explain that right now off the top of your head? Yes. But I want you, you will get it on that same page. We'll talk about it. Draw and label both plasmids. Where can you find those? B12. I tell you exactly where to find everything. This should be 100%. Be sure to include all the components of each plasmid. Did we already say what each part does? Yeah, you have it in your notes. Okay. So note right here, it says start a new paragraph for each lab. So 3 paragraphs total. Do not write as if answering questions. Just use the prompts below. Okay, so you need to write it in complete sentences, put in your drawings where needed. So this would be number 1. Second paragraph, from lab 3, explain the value of sticky ends. Do you know what sticky ends are? We gone over that? Yes, and the role of ligase in genetic engineering. You will find that on page B25. From lab 3, draw and label the recombinant plasmid you hope to form. That is found on page B27. This is maybe a 1 sentence and 1 drawing, and that's it for this, right? Maybe 3 sentences, actually 2 sentences up here and 2 drawings. Right here, for lab 4, explain the value and process of gel electrophoresis. Do you already know about gel electrophoresis? Yes, but get it from B38 and B41. As far as your hypothesis is, I have already written that for you. Identified your variables, your controls, and your constants. All you have to do is put it in your notebook. Should you get 100% on that? Yes. Okay. Now, if you look up here, the flow chart, because you don't have any data until you do gel electrophoresis, so you can do the flow chart as we go. If you want to do it all ahead of time, be my guest, because you have all the resources for it. Right here it says you need to have everything up, what we've talked about here, all the way through for lab 2 by 328. Draw table 2.2 here for your flow chart. That is found on page B15. And indicate that you will do step 6, found on B16, with very simple diagrams. That's what has to be in on Tuesday. This needs to be in by 3:30, and this one needs to be by April 3rd. But look right here. See sample pics inserted at the end of the document to help you with your diagram. So let's go to the end. Oh, here is my flow chart for lab 2. That might be helpful. Okay, you can come up with your own, but if you want to use this to assist you, here it is. And here's that data table I told you about. Follow the table. Okay, so that's my expectation. And that's as far as you, hang on just a minute, you guys, as far as you need to be by Tuesday as to here, then you add the rest of the flow chart by that day and the rest of the flow chart, and you don't need to do that until the days that we're actually doing the lab. Okay, got it? Okay, see you Friday with the first one done.